Hello everyone. Today I'll be presenting the COGGNN, Cognitive Graph Neural Networks in Generative Connectomics. Generative AI has transformed so many fields such as drug discovery, NLP, computer vision, etc. In neuroscience, a nascent field called generative connectomics is emerging with applications such as graph super-resolution, brain evolution prediction, cross-modality graph synthesis, and CBT learning. The backbone of these advances has been graph neural networks, but they focus only on preserving the structure and topology of the connectome while they overlook a key feature of the brain, which is cognition. This creates a big gap. We need generative models that capture both structure and cognitive capacities. So I proposed a solution called COGGNN, which is a first cognitively enhanced generative GNN, where it integrates a cognitive reservoir, which is a brain-inspired memory mechanism, to the generative process. COGGNN is general. It can integrate multiple cognitive traits such as visual, auditory, language, and can be applied to different applications of the generative connectomics. In our work, our proof of concept is VIS-COGGNN, where we will integrate visual memory for CBT learning. CBT is, or connectional brain template, is an average representation of a population of brain networks. So here, the cognitive input used is visual, and we will try to generate a cognitively enhanced population level brain template. Our method takes as input training subjects, where each subject is represented by multi-view brain networks, which are fed to a GNN. The GNN learns embeddings for each brain region, and from these embeddings we apply tensor operations to compute subject-biased CBT. This CBT is then instantiated as a cognitive reservoir, and visual sequences are projected into the reservoir and processed through its recurrent weights, where the task here is to recall delayed versions of the input images. This mimics how short-term memory works in the brain. To guide the learning process, we perform a co-optimization with two objectives: a centeredness loss that ensures the CBT remains representative of the population, and a cognitive loss that enforces successful recall of delayed inputs, which strengthens the memory-like traits. Together, this dynamic feedback loop enhances both the CBT structural representativeness and its visual memory capacity. Finally, after the co-optimization, we refine the results by aggregating all subject-specific CBTs with a median operation. This produces a final population-level CBT that is symmetric and cognitively enhanced. For evaluation, we use two datasets, the Alzheimer dataset and the autism dataset, where we used 4 cortical features for 77 subjects in AD and 6 cortical features for around 310 subjects. The ROIs are 35 from Disick and Kilvaney Atlas, and we analyze it in both cases, the left and right hemispheres, separately. For training, we built separate models per hemisphere across the 4 groups, and we used 5-fold cross-validation using Adam optimizer for optimization and 500 epochs, and we used early stopping to prevent overfitting. For the visual input, we used the MNIST dataset of size 10 by 10, and the time lags range from 5 to 40. And as a benchmark method, we compared against the deep graph normalizer DGM, which is a strong state-of-the-art method for CBT generation. In the results, to assess the centeredness of the generated CBT, we compute the mean Frobenius distance, which measures the average deviation between the generated CBT-C and the different network views AK in the testing set. Across datasets and hemispheres, the VIS-COGGNN consistently achieves lower distances than the baseline DGN, where it outperforms in about 75% of datasets. Then we evaluate the visual memory capacity of the generated CBTs by DGN and VIS-COGGNN. We use VIS-MC, which quantifies how well the system retains and reconstructs visual inputs over time by summing the squared correlations between the delayed inputs and the predicted outputs across multiple time lags. As shown in the plots, the VIS-COGGNN significantly outperforms DGN, achieving statistical significance results in both AD and ASD datasets. These results underscore the benefit of integrating cognitive mechanisms into the CBT generation, such as visual memory, which is the key feature of our approach. To conclude, we propose the first cognitively grounded generative model for brain networks, which opens a new direction for biologically inspired connectomics. Clinically, the cognitive CBT may serve as biomarkers for brain disorders such as Alzheimer's and ASD. For future work, we aim to extend to other cognitive domains such as auditory language and to different other applications in the generative connectomics. To learn more about this aspect of cognitive computing, you are welcome to visit our YouTube video and visit our poster. Thank you.